Aloha everyone, and welcome to today's lecture where we are going to begin Unit 3 of the material. So we're already halfway through this course. We're going to start talking a little bit about DNA. So I mentioned DNA in Unit 1, but we're going to talk much more in depth about DNA today. We're going to look at DNA's structure, and then we are also going to talk about DNA replication. And this all goes along with Chapter 7 in your e-text. For today's learning objectives, first you should be able to define the structure of DNA. So what exactly is it composed of, what does it look like, and how exactly it is organized into our cells. Secondly, you should learn how DNA is copied, both naturally in our living cells, and also how we are able to amplify it for forensic work, which is kind of what this whole chapter in your e-text is about. And then finally, I want you to understand the concepts of polymerase chain reaction, or PCR, as well as DNA profiling. So we're going to talk about all three of these learning objectives, and by the end of this lecture, you should be able to answer each of these questions, kind of have a better understanding of these learning objectives. So let's go ahead and get started. Here is a little bit of review from Unit 1. So nucleic acids, if you remember, are one of the 4 types of macromolecules. So we had our carbohydrates, proteins, lipids, and then nucleic acids. Nucleic acids are responsible for transmitting genetic information. So in other words, what makes you, you? The subunits of nucleic acids are going to be your nucleotides. So just like carbohydrates had different types of monosaccharides or disaccharides, proteins have amino acids, nucleic acids are going to have nucleotides. And there are 2 main types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. So in this lecture today, we are going to solely focus on deoxyribonucleic acid, or DNA. But throughout this Unit 3, we will be talking about RNA as well. So DNA, like I said, we'll be focusing on that today. DNA is the molecule of heredity. So it's going to provide the instructions for making proteins. And again, DNA is really what makes you, you. Nobody has your exact same DNA unless you have an identical twin. So DNA, again, stands for deoxyribonucleic acid. This is a hereditary molecule that is passed from parents to offspring, and DNA is common to all living organisms. It serves essentially as kind of the instruction manual for how to build an individual. So if you think of back to maybe when you were a kid and you got a box of Legos or something that you had to assemble as a toy, right? It came with instructions, and those instructions told you exactly how to build that toy. Same thing here. Our DNA is essentially the instruction manual for how we are built as individuals. DNA is found in the nucleus of eukaryotic cells. So again, take yourself back to Unit 2, where we talked about eukaryotic versus prokaryotic, right? So prokaryotes, they have the DNA just kind of floating around inside of the cytoplasm, whereas in eukaryotic organisms, such as ourselves, that DNA is going to be found inside the nucleus. And you'll notice as we kind of go through Unit 3, we're going to be mostly focusing on eukaryotic organisms. So I want you to think of that DNA as being housed inside the nucleus, or that control center of your cell. DNA is found in the nucleus in a certain form. So we usually think of DNA as just being this stringy thing, right? But within the nucleus, it's actually found in the form of a chromosome. So a chromosome is a single DNA molecule that is wrapped around proteins. So they're actually called histone proteins. So if you look over here, you see your nucleus. You have inside that cell these chromosomes. This is a chromosome. Essentially, it looks like a big kind of bolded X, right? And it's within this chromosome, that DNA is just very tightly wound, and it's wound around proteins. So chromosomes are made up of long strands of DNA, and they're tightly coiled around proteins called histones. And this creates the compact structure that you see right here in this figure. So kind of take away that notion that DNA just looks like this little stringy mess, right? Inside the nucleus, it's in the shape of these chromosomes. And that will kind of help you as we continue to go through Unit 3, because we'll be talking about things like mitosis and meiosis. And you're going to see figures where they're in this chromosome shape. So that's why I kind of wanted to introduce that to you here, so you kind of get it in your head a little bit that, hey, they're in this chromosome shape. And I think that will help you when we cover the processes of mitosis and meiosis. Talking a little bit more about those chromosomes, humans actually have 23 pairs of chromosomes. So if you think 23 pairs, that means 23 times 2, we have a total of 46 chromosomes. But we usually talk about them in pairs because one chromosome from each pair is going to be inherited from your biological mother, and then the other pair is from your biological father. So you can see that here in this figure. This one's actually from your textbook. So they're just looking at the set of chromosomes 3, right? You have 23 pairs total, right? 23 times 2 gives you 46. So if you look here, you can see one of these chromosomes comes from one parent, so say your mom. One comes from the other parent, say your dad. So you have a total of 2 chromosomes for each one of these pairs. So 23 pairs, 46 total. And this is going to be different depending on what animal or what species you are talking about, right? But I just want you to know the number for humans right now. So 46 total chromosomes. You can see here 23 total pairs. The 23rd pair of chromosomes is a little bit different because it is that 23rd pair, that very last pair, that determines the sex of the individual. So in other words, sex meaning female or male. So if you have 2 X chromosomes on that 23rd pair, then sex-wise it is female. XY would be male. So you can see that here, this is that 23rd pair. If there were 2 X's, this individual's sex would be female. If there, as you see here, is an X and a Y, that individual sex is a male. So something that I want you to keep in mind, especially if any of you are like history buffs and you think back to the time of some of the different kings that would get really mad at the queens for not giving them a son, it is actually the dad that is going to be determining the sex of the individuals. So if you think about it, the mom can only pass on an X chromosome, right? Because each one of those pairs is going to come from one of the parents, right? So a chromosome from one parent for the 23rd pair, the female could only give an X. No other choice, right? The male, on the other hand, can give either the X or the Y. So it is actually the male that determines the sex. So maybe you'll get some trivia questions sometime about that, or if you are a history major or something like that and reading about different previous queens and stuff back in a long time ago, back when it was, I mean, some were sentenced to death for not even giving the king a son. So kind of crazy considering that it is the male determining the sex of the offspring. Now we're going to look a little bit more at the actual structure of DNA. So DNA, like I said, is composed of nucleotides. But what exactly is a nucleotide? So a nucleotide is going to have 3 things that compose it. A nucleotide is composed of a sugar, a phosphate, and a base. So you can see over here in this figure, your deoxyribose sugar, which is exactly where we get deoxyribonucleic acid, right? Deoxyribose gives you that first part of deoxyribo, right? So this is what we call a pentose sugar. If you think of it, it looks like a pentagon, right? So it is a 5-carbon sugar. And this is one part of that nucleotide. The second part is going to be the phosphate. We talked about phosphates in Unit 2 when we were talking about ATP and ADP, right? The phosphate groups. And then you have a base. Sometimes I've noticed that students tend to have this, they tend to think that the base is the only part of a nucleotide. So they forget about the sugar and the phosphate and they just talk about the bases. But that's not correct. The base is actually one of 3 parts of that nucleotide. So what happens is the phosphate and the sugar, so these 2 portions right here, form kind of the backbone of DNA. And then those 4 bases are going to be in the middle. So the 4 bases are adenine. These are nitrogenous bases. You have adenine, which is abbreviated as A, guanine, abbreviated as G, thymine, abbreviated as T, and cytosine, abbreviated as C. So you have 2 strands of nucleotides in DNA that are going to pair up. And essentially they twist around one another to form that spiral-shaped double helix, right? That you see right here in the background of the slide. This is a double helix shape, right? So what happens to make this shape of DNA is that the sugar and the phosphate are going to form the outside. So this outside portion that you see here, same thing here, this outside portion here, this outside portion here, that is going to be essentially the backbone, the sugar-phosphate backbone of your DNA molecule. Something to note is that the phosphates and the sugars are actually covalently bonded together. And if you think back to when we talked about covalent bonds, I mentioned they're very strong bonds, right? And that's exactly what we want, right? We're not going to want this backbone of our DNA to be broken. We don't want that. So it's important to note the sugar-phosphate backbone here is covalently bonded together. It is very strong. Then your bases form essentially the rungs of the ladder, right? So if you look in the background picture here, they look like little rungs that you can kind of climb up on a ladder. They're the internal part of that molecule. And they are linked together not by covalent bonds, but by hydrogen bonds. So those hydrogen bonds, we talked about hydrogen bonding in depth when we looked at water and the different properties of water, right? Hydrogen bonds are weaker. And it's important that these bases are held together by these hydrogen bonds because those hydrogen bonds can be broken. We need them to be broken so that that DNA molecule can be essentially unzipped when we do the process of DNA replication that we'll be talking about in just a little bit. So if we look more at DNA structure, I mentioned to you that there are 4 different nitrogenous bases. You have cytosine, guanine, thymine, and adenine for DNA. These bases have what we term complementary base pairing. And what I mean by that is that adenine and thymine are always going to be paired together. So you'll always see adenine pairing with thymine, having those hydrogen bonds between, and they are going to be held together by 2 hydrogen bonds. So you can see over here in this figure, adenine in light yellow, thymine in green, they're always going to pair together, right? Then you have guanine and cytosine. They have 3 hydrogen bonds between them, but you will always see it being guanine and cytosine that pair up together. You can see that over here, guanine in red, cytosine in orange. They are always pairing together. So when you have 2 of these nitrogenous bases together, like you see here, you have cytosine and guanine, that together is called a base pair. So that's why we term it complementary base pairing. A always pairs with T, C always pairs with G. And again, you have those 2 strands of that double helix DNA being held together by this base pairing. And this is done through hydrogen bonding. So this pairing actually allows for DNA to replicate itself, which we'll see when we cover the process of DNA replication. What's really cool is that the specific kind of sequence of nucleotides along a strand of DNA is unique to each individual. So the order of the bases here, so you could have adenine, thymine, the order of these different bases, so if you were to read kind of down starting from here, you have guanine, adenine, cytosine, thymine, so that is going to be different for each individual, right? Different parts of your DNA are different, and that's what gives you your individual characteristics. That's also why kind of the whole chapter in your e-text focuses very heavily on the use of DNA in the judicial system now, right? Because that DNA can kind of be the key to either convicting someone of a crime or exonerating people that were put away for a crime years ago when we didn't have the advanced research that we have about DNA. We weren't able to amplify the DNA found at a crime scene like we are now. So again, I just want you to make sure you really understand the structure of DNA. DNA is a nucleic acid. It is composed of nucleotides. Nucleotides have three parts to them. They have that sugar, they have the phosphate, and they have the nitrogenous base. For DNA, that sugar is deoxyribose sugar, and that, along with the phosphate, makes this backbone, this kind of blue ribbon-like structure. That is the sugar-phosphate backbone. Then you have four bases in DNA. You have adenine and thymine that always pair together, and they have two hydrogen bonds between them. And then you have guanine and cytosine that are always going to pair together. They have three hydrogen bonds between them. So it's said to be a little bit stronger of a bond. There might be some mnemonic devices to kind of help you learn this. The only thing I've heard is if you look at these four bases and figure out which one makes a word, then that will help you. So ag doesn't make a word, AC doesn't make a word, AT makes the word act. So adenine and thymine go together, that would leave you with guanine and cytosine going together. So if that helps you remember that, maybe there are, if you Google, you might be able to find some mnemonic devices. But just make sure you really understand the structure of the DNA molecule. We're now going to move into our second learning objective, which is talking all about DNA replication. So DNA replication is the natural process by which cells make an identical copy of a DNA molecule. This takes advantage of that complementary base pairing, those rules that we were just talking about on the previous slide, right? Adenine pairs with thymine, cytosine pairs with guanine. So when we look through, kind of talk about the different steps of DNA replication, I'm going to give you a very kind of basic how exactly this works. There are some great animations in your e-text. There are also videos you can watch online if you want, but you should note that some of those videos are going to be much more complicated than what you need for this class. Kind of the same thing that I talked about with aerobic respiration, right? You all are not science majors. I want you to have the basics of it, be able to kind of apply it to your life. You don't need to know the nitty-gritty details that you just have to memorize and then forget for an exam, right? So we're going to kind of walk through the basics of the process of DNA replication. So first, what has to happen is that the hydrogen bonds that hold the base pairs together, so remember there are three of those holding together cytosine and guanine, two of those holding together adenine and thymine, so those have to be broken. And in order for this to happen, we have this enzyme here in purple. This enzyme is helicase, and it's an enzyme that essentially unwinds the DNA double helix. So what happens, you can see you have your DNA, it looks normal here, right? Then that helicase enzyme is going to bind, and essentially it unzips the DNA. So think about a zipper on a jacket, right? Think of that kind of being the two strands of DNA, that double helix held together. Then you take that zipper and you pull down on it and it starts to unzip. So that's exactly what's happening here, is unzipping of that DNA. You can see those bases are no longer paired together. So the helicase enzyme is essential. It's going to bind to the DNA, and then break those hydrogen bonds, unzipping the DNA strand. After that DNA molecule has been unzipped, the next kind of step here is that we have to add in more nucleotides, right, to make identical strands of DNA. So next you have the DNA polymerase, shown here in green, right? This is another enzyme, and DNA polymerase reads the DNA, and what it does is it adds complementary nucleotides, complementary bases to that, and as well as that sugar phosphate backbone, so the whole nucleotide, using the rules of base pairing. Again, A goes with T, C goes with G, right? And again, DNA polymerase is another enzyme here. So helicase first comes in, unzips it, and then DNA polymerase is going to go and is going to say, okay, this one is adenine, I need to add a nucleotide with thymine. The next one is guanine, I need to add a nucleotide with cytosine. So essentially, it's remaking an identical copy of that DNA so that in the end, you have double the amount of DNA. And that's kind of the process of DNA replication, kind of put very simply, right? Helicase comes in, binds, it breaks apart those hydrogen bonds, kind of unzipping that DNA strand. Then you have the DNA polymerase. It's going to bind on both of those unzipped strands, right? And then it's going to read what's in that DNA strand and put a complementary nucleotide there. So in the end, you end up with double that amount of DNA. In terms of this process of DNA replication, it is what we call a semi-conservative mechanism. And what I mean by this is that DNA replication produces two copies of that original DNA molecule. Each molecule is going to consist of one of the strands of the original DNA molecule. Sometimes we term that the parent molecule, and a new strand, or sometimes we call that a daughter strand. So you can see over here, again, this is semi-conservative replication. We have our original parent strand right here. It's in this dark kind of bluish purple. We go through DNA replication once, and then we can see that there's one purple strand here, one purple strand here, but a pink one was added and a pink one was added. So in other words, in this DNA here, we have one of the original parent, one of the daughter strands, right? So it's semi-conservative. We're not starting from scratch. We're not keeping the exact same thing. It's semi-conservative. We have one of those parent strands, one new strand. One parent strand, one new strand, right? That's what's meant by semi-conservative mechanism. So with this DNA replication, sometimes students kind of wonder, okay, like, why are we even talking about this? Like, we have our cells. Why do we need more DNA? Well, do your cells last forever? Think, for example, your skin cells, right? How many times have you been out in the water or taken a shower and then some of your skin sloughs off, right? So those are skin cells. Those skin cells have DNA in them, right? How many times has your hair fallen out? Your hair has DNA in it. So we don't keep the cells that we're born with, right? We're constantly kind of making new cells for at least certain parts of our body. Brain cells, that's a little bit different. Spinal nerve cells, things like that. There are some cells that cannot regenerate, but a lot of our cells are going to die. They go through something called apoptosis, which is cell death, and we need new cells. So in order to make new cells, we need DNA, right? So we need this process of DNA replication in order to make new cells. So with our DNA, each person's DNA is unique, as I mentioned, and we can actually extract DNA from cells. Maybe you've done a lab where you take like a toothpick and you rub the side of your cheek cell on the inside to get some cheek cells to stain. You could also send that in. Maybe you've heard of 23andMe, things like that. Maybe you've done an ancestry thing where you had to scrape some DNA off, right? So what happens from that is when we extract DNA from cells, we create what we term an individual's DNA profile. This gives us a visual representation of a person's unique DNA sequence. And to make a DNA profile, we may need to take that little sample of DNA that we have and make many copies of it. To learn how to make copies of DNA, we had to first learn this process of DNA replication. So now we're going to kind of move on from talking about what happens in our bodies for DNA replication, and we're going to talk about things like polymerase chain reaction, where we can do this DNA replication process, but we can do it in a tiny little test tube. Our last learning objective for today is to have an understanding of polymerase chain reaction, or PCR, and also DNA profiling, and how DNA profiling can be really useful for the judicial system. So PCR is a laboratory technique that scientists use to amplify just fancy term for replicate, a specific DNA segment in order to study it. In order to do this, we need different components, and we learned what components we needed by understanding the natural process of DNA replication that we just covered. So the different things that you're going to place inside of that test tube are obviously your DNA sample that you want to amplify, free nucleotides, because you need those to be added to the DNA strands, right? And of course, we need that ever important polymerase enzyme to add those nucleotides. And we also need primers. Primers are very short segments of DNA that are going to guide that DNA polymerase to the section of DNA that we want to copy. There are also some other things you can add in here. If any of you have done PCR before or heard about it, probably heard of something called TAC. There are some other things. I did a lot of PCR in an undergraduate internship in South Carolina, and it was very interesting working with benthic microalgae, and we were trying to figure out the community there. So what we would do is we would take that DNA, we would first have to extract the DNA from the sediment, the sand, and then we take, because there was such a small amount of DNA in there, we had to go through this process of PCR to try to amplify that DNA so we could figure out what organisms were living there. Right? So same thing that you would do. Your textbook, again, is focusing all on kind of the judicial system and trying to either indict people for criminal activity or trying to exonerate them. So say that a crime scene occurred. Generally, you're not going to find a lot of DNA. Like, unless somebody just did a really bad job of pulling off a crime, you're maybe going to find the tiniest little amount of skin. Or maybe you find just like part of a hair, something really small, all right? And so you're going to take that tiny little amount of DNA and you want to go through this process of PCR to make a lot of copies. So in this process of PCR, you're going to add all these ingredients into your little test tube, right? What happens then is you put these little test tubes in a machine to do this process of PCR. So first of all, the machine is going to heat up those test tubes. It heats them to separate the DNA strands. So heating separates the DNA strands. It does the exact same thing that helicase naturally does in our cells, but we don't have helicase, right? So what we're doing instead is we heat it, and it does what we call denaturing. So we know that extreme heat can kind of damage cells, right? We know that just based on all the information we've heard about skin cancer, right? So same thing, heat will actually take and it separates those DNA strands. Then after going through this heating process, we see they've been separated, right? Then the cooling, the machine stops heating and it starts to cool. That allows DNA polymerase, that enzyme, to pair the new nucleotides with the original template strands. And then you go from one strand to having now a total of 2 strands. And you keep doing this over and over again. The machine you can set for the number of replications that this cycle is done. So kind of the end result of PCR is it's allowing DNA replication to occur many times. So from a starting sample of just a few DNA molecules, PCR can actually make billions of copies of a specific region of DNA in just less than a few hours. So it is a very cool process. It was kind of big, just starting to be big, I guess, when I was in high school. We've learned even more about it. We can do even more with it now, and I'm sure in the future, you know, the world is kind of our oyster as we get more technology, kind of learn more about these processes. But again, very cool process. Maybe someday it might be something that you have some sort of experience with. I hope that it's never for any of you going to commit a crime, but you know, maybe like you hear or you read in the newspaper about somebody being exonerated of a crime because they were able to take that DNA sample and replicate it and say this is not that person's DNA. So that's kind of some way that you can apply this, I guess, because you may hear about something like this and be like, oh, I remember in BIOL 101, we kind of talked about this process of polymerase chain reaction. The other process that I want you to have just kind of a basic understanding of is what we term DNA profiling. DNA profiling actually allows us to identify the unique characteristics in the DNA of a person. So each person's DNA sequence is unique, right? And it can be used as kind of a molecular fingerprint. Everyone has a genome, the human genome. You probably heard of that, the Human Genome Project. So genome is just a complete set of genetic instructions that encode in all the chromosomes of an organism. So you have your genome, I have my genome. Everybody has their own kind of unique genome. Determining the sequence of the entire genome is expensive and unnecessary. So what happens is essentially we take a shortcut with DNA profiling, and we just analyze a portion of a genome when we're doing these DNA profiling cases where we're trying to take that DNA out of scene. We've already amplified it. How can we match that to different individuals to either figure out if this person's guilty or if this person never should have been sent to jail 15 years ago? So essentially we're going to take a shortcut, and instead of analyzing the entire genome, we're going to look at specific portions of the DNA. The specific portions that we look at of that DNA are what we term STRs. These are short tandem repeats. These are a good type of DNA to actually use for DNA profiling. And we can use PCR. If you remember, I talked about those primers that tell that polymerase, the DNA polymerase, where exactly to bind. So we can use those primers to designate, hey, I want this STR to amplify a bunch of times. And so we can use that PCR to target the STRs. STRs are just sections of a chromosome in which short DNA sequences are repeated. So for example, the sequence AGC may be repeated over and over again. And the number of repeats are going to vary from one person to another person. So say that sequence AGC, I have that repeated 10 times. You might have it repeated 8 times or 6 times or 4 times. So those STRs are in the same places along everybody's chromosomes, but they're repeated a different number of times in each individual. So the length of that STR region is going to vary from person to person. And this allows us to essentially fingerprint people based on their DNA. So here's a figure that was shown in your e-text. So say you have this person here. You look at this STR region, and then you have this person here, this person here. So this STR region, you can see it's repeated 4 times. This person is repeated 6 times. This person is repeated 8 times, right? Whereas this top STR region repeated 6 times, then just 3 times, then just 2 times. So again, those regions are going to be, the length of that varies from person to person, and that allows us to do this DNA profiling without analyzing the entire genome. We just take these certain little STR segments that we're able to focus on. If we look just kind of basically at this process of DNA profiling, what first happens is we need to collect cells and extract the DNA. So maybe somebody's going into the crime scene and they see a little bit of like a fingerprint that might have a skin cell on it. Or maybe they find a small piece of hair. So that's going to be collected, and we have to go through this process of DNA extraction to extract that DNA from that sample. And that's a whole nother process. We're not going to get into that. But when you're doing this first process of collecting the cells and extracting the DNA, we have to be very careful not to contaminate the samples with DNA from other people, right? So that's why always gloves should be worn, right? Very carefully moving things, into the sample collection vial so that you're not contaminating your sample. Next, we want to amplify the STR regions by this process of PCR. The more regions that we amplify, the more certain we are about the results. So maybe we don't have a lot of time, don't have a lot of money, and we just do one region. Well, we're not going to be super confident in our result because it's less likely that we are correct. Same thing if back when we talked in chapter 1 about going out and doing an experiment. If you have just a couple of people in that experiment, the results aren't going to be that great, right? The more people you have, the better your data is going to be. Same thing here. The more regions we amplify, the more certain we are that we actually have a correct result. The next step then is to separate those STRs using the process of gel electrophoresis. This is a laboratory technique that separates the DNA fragments by size. And this may be something, I'm not sure if you actually still do this in lab for this class. I know it used to be done. But this gel electrophoresis is essentially this, it's usually kind of this clear acrylic, almost looks like a Tupperware container essentially that has electrodes on it. And then you have an agar gel. It kind of looks like clear jello that's generally in the shape of a rectangle. And on one end, you have these little wells that you're going to load the DNA into. So you can see that here. Think of this as like your agar gel. Here are these wells. That's where you load your STRs into. And then because DNA is a very heavy molecule, DNA has a negative charge. So you always set up your gel electrophoresis machine so that you have the negative end where you're inputting your little STRs, and then positive is on the other end. Just like a battery has a positive and a negative end, same thing here. So there's an electrical current on this gel once you start it, and DNA has a negative charge. So it wants to go towards the positive end, right? So this generally on your gel electrophoresis little machine, you'll notice that the black end is usually the negative end, and then the positive end is usually the red end, but it just kind of depends on what machines you're using. So what happens is you load all these, you turn the machine on so that electric current starts, and the smaller fragments are going to travel further because they weigh less, right? And the larger fragments are going to stay closer. But essentially, we're separating out these STRs, right? And it's going to leave distinct bands where each one of those separated out, and that is going to allow us to do this process of DNA profiling. After that process of gel electrophoresis has finished, then we're going to take and compare that STR banding pattern. So you can see down here the different bands, and usually the bands of these STRs are visible using fluorescence. So at least at the lab that I worked in, there was kind of this little hooded area where you had a fluorescent light. You'd put the gel in there, you'd close the door, and then on the computer screen it would pop up and you could actually view that, take pictures of it, zoom in to see those distinct bands. So right here you have an example of one of these gels. In this first well is the cheek sample, then suspect A, suspect B, suspect C. So what you're going to do when you're doing this in terms of DNA profiling, trying to either convict or exonerate somebody for a crime, you look and you see you want to compare everything to this DNA sample that you found. So look at this first blue band. Suspect A has that blue band, but they have two red bands right by it. That immediately tells us suspect A is not the person who committed the crime. Suspect B has that blue band. Suspect C has that blue band as well. Then we move on to the next band. You have this red band here. You do not have that same red band here for suspect B. Instead, you have two smaller red bands further apart. Suspect C, however, has that exact same red band. So far, suspect C still matches up. You then continue on to the blue, matches, green, matches, right? So all of these STRs match perfectly with suspect C. They are a perfect match here. So the different lengths for each STR region and each person create this specific pattern of bands. So again, when visualized with a detection technique known as fluorescent, this is the same type of light that you see released from a glowing jellyfish. The separated segments of DNA create a specific pattern of bands that is unique to every single person. And this pattern is the person's DNA profile. Scientists can compare the bands of DNA from a crime scene to DNA from a suspect. And DNA profiles obviously have other applications as well, such as paternity and ancestry testing. So 23andMe, other things like that. If you're ever wondering what exactly happens with that cheek cell that you turn in, that's exactly what they're doing, right? They first go through the process of PCR, then they're doing this DNA profiling. So as I mentioned with DNA profiling, we want to look at multiple STR regions, because when we use many regions, the chance of any other person sharing that exact same profile becomes impossibly small. And this is shown in your textbook. So if you were to use one STR region, the people that share that is 1 in 5. So 1 in every 5 people are going to have that exact same STR region. If you do 2 STR regions, it becomes 1 in 25. 5 STR regions, 1 in 3,125 people. If you do 15 STR regions, it's 1 in several quintillion people. So you can see really quickly it increases, right? In terms of it's much less likely that you're going to have the wrong individual. Say if you're using this for a crime scene and trying to match suspects up with it. So the likelihood that any 2 unrelated people will have the same number of repeated sequences at all 15 regions is 1 in some number of quintillions. So billions of billions is what's meant by quintillions. So DNA evidence is much more reliable than other forms of evidence. For example, your textbook talks about a prisoner that was put away due to bite mark identification. The error rates for that can be as high as 91%. Hair analysis can only exclude a suspect. It can't positively identify one. But when we look at DNA, unless you have an identical twin, no 2 people share the exact same DNA. So this is a really good mechanism to be used in the judicial system, right? This is also very useful. Like I said, we were using PCR and gel electrophoresis to try to figure out different species, right? Because each species is going to have a distinct genetic pattern as well, right? So it's also very useful in the biological field just for doing research on different species that might be living in different areas. For like, I was working with benthic microalgae, so microscopic, right? You wouldn't know. We would look at that same kind of thing in the digestive, like the fecal matter of organisms that live there, to try to figure out what those organisms were eating, what species of benthic microalgae were the best food source for them. So lots of different applications for PCR and DNA profiling. That is all for this lecture on Chapter 7 in your e-text. Again, it's a little bit shorter of a lecture, but your other lecture this week will be a little more intense, complex than this one. So again, to-do list, just make sure you read or at least skim through Chapter 7 on Achieve. Complete the Chapter 7 adaptive quiz on Achieve. Again, that's all or none. So you can get all the points there. And then complete class activity number 6, just really short, but making sure you understand the structure of DNA and how DNA replication works. That's all for this lecture. Mahalo.